special edition of our discussion group. It's 3:30 now, so I can start. So it's my pleasure to introduce Michael Dymock. He's a biostatistician based at the Telethon Kids Institute in Perth, Australia. He's doing a PhD program at the University of Western Australia. He's supervised by Dr. Julie Marsh and Professor Tom Snelling, and further advised by Rob Mahar and Professor Kate Lee. He's also part of Austream, which for those that know TMRP is kind of the Australian analogous. And he's interested in designing innovative trials that better align with the requirements of decision makers, specifically vaccine policymakers. So with that being said, the floor is yours, and you will present us this talk on designing Bayesian clinical trials to enhance vaccine policy recommendations. Thank you.
Thank you very much, Sophia, and thank you all here and online. It's certainly a great honour to be invited to talk about my work to you all. Just a little bit about myself to start. Sophia gave a great summary about that. Telethon Kids, funnily enough, changed their name about 3 weeks ago or something. So we're now called the Kids Research Institute Australia. They took away all my polo shirts, so that's quite unfortunate. I've got some more information on my website there. Presumably, I'll be able to share the slides with everyone if anyone's interested in that. I've also got some hyperlinks throughout to different resources. A bit of motivation about my work. So I've been working for just over 4 years as a clinical trial statistician. It's been great. There's been lots of opportunities and challenges. And then about a year ago, I started my PhD. And in that, I was trying to address what I see as possibly a fundamental problem in the way that we design clinical trials. So for an outline of my talk, I'm trying to sort of make an argument from first principles. I'll probably go over a little bit of some content that everyone's probably quite familiar with. I'll then move into vaccine policy recommendations in Australia and describe how that is done, and then I'll provide a potential solution for that. This is certainly a work in progress. As I said, I'm 1 year into my PhD program, and so I've got about 2 years left, and this is probably quite a pivotal moment where I can sort of choose the direction to finish off in. So from first principles, the way that I think that we do clinical research is I think we do it to broadly improve human flourishing. There's I think it's the mission or the vision statement from the Kids Research Institute here. The idea is basically we do research to improve health outcomes. The way that we actually get there is by improving healthcare policy and practice. And the way that we get there is by improving the way that we make decision-making, the way that we make decisions within a healthcare context. And it's really this box here that I'm particularly interested in. And I think to at least some extent, we argue that this is why we do things like clinical trials, is where we're trying to make better decisions. So is there a problem? Well, I think most people can anecdotally say that results from clinical research only slowly moves into policy and practice. Often we're constrained with our resources, and so we do a clinical research project. We then publish it in the literature somewhere. We then hope that makes an impact somewhere down the line, but often as statisticians, we've sort of moved on and we're doing other sorts of projects. There are many reasons for this. Some of them are statistical, and that's what I'm going to try to address. I could only dream of approaching all of the other issues. Part of this reason is because I think that we usually design research to register or to approve a drug or a vaccine, but it should prioritise the decision making of our stakeholders, which are consumers, clinicians, and policymakers. I've got a little asterisk there for should, because I don't necessarily think all clinical research needs to fit into this basket, but especially if we're publicly funded and if in a grant application we're saying the objective of this research is to inform policy and practice, then that is what we should be trying to do. Just a little note on hypothesis testing. Everyone will be quite familiar with this point, but what we're really doing is we have some usually complex scientific hypothesis about some biological phenomena. We're distilling that into a statistical hypothesis where maybe we have a parameter of interest and we're particularly interested in the value that parameter of interest may take. So for example, here you could say this treatment affects beta. It has no effect or a negative effect, or it's got a positive effect. So one thing that we do is distilling the complexity of the scientific hypothesis into a statistical one. But then something that I guess I'm concerned with is, if we say claimed hypothesis B was true, we then make the assumption that that means we should therefore recommend the treatment, that we should then therefore implement this treatment in practice. But of course, that there's a bit of a conceptual jump there, because the value of this parameter, it's quite possibly measuring one component, where a policymaker is going to consider a whole raft of different issues. And so as scientists, what we like to do is make some declaration about this hypothesis is true, or I don't have evidence to claim that hypothesis is true. The second we do this, we're making a truth claim, and we may be wrong. So that's how you induce these concepts such as power and type 1 error. I think as Bayesians who design clinical trials, especially say if I'm designing an adaptive trial, we often get into the habit of saying, and here are the frequentist operating characteristics of my design. But I don't think it's necessarily frequentist, because as a Bayesian, I might show you a posterior distribution. I might summarize it with a credible interval, might make a probabilistic statement such as the probability beta is positive is 98%. That's fine. But the second I then say, and therefore the treatment is superior, I'm now making a claim. And the second I make that claim, I then get into the type 1 and type 2 error realm. So traditionally, we'll have a primary analysis. Why do we do that? Because we want to have some sort of control over these errors. So for example, we could have a treatment effect beta. We set a decision rule. We do that to control this type 1 error and power. We then have, using that decision rule, we'll make some declaration about if the treatment is effective. But then we enter the trial and we report on all outcomes. Now, the way that I see it is that everything sort of surrounds this hypothesis test. The way we design decision rules to suit the hypothesis test, we, at least in theory, choose our sample size based on the hypothesis test. Of course, in practice, sometimes the sample size is determined by funding constraints, and then we do this awkward sort of backwards power calculation to determine the power. And quite often we'll have, say, clinicians think that the trial is only successful if we declare the treatment superior, and otherwise we haven't learned anything. But in practice, so in reality, we've collected all of this data. We've progressed from a prior state to, say, a posterior state. We've got an updated sense of knowledge of the world, and so we should be able to use that in a more sophisticated manner. And then, unfortunately, historically, this is how we've chosen to publish. Of course, I think we're, as a field, moving past that. So, does it suit clinical research? Well, if we're looking at healthcare decision making and improving it, sometimes this isn't going to be the perfect answer, because the decision maker usually looks at multiple outcomes. They look at efficacy, they look at safety, they care about the effect size, they care about how much uncertainty we have in that particular effect that we're estimating. So this single effect measure into a primary outcome, into a single declaration, may not suit us perfectly. I'd like to also just make a little note on what I consider the pursuit of truth and the role of science. I view science as a tool that we use to uncover the truth about various phenomena in the universe. I think it's a good thing to do science and to understand that phenomena. I think if we do so, it'll lead to improved healthcare and human flourishing. So all of this, I'm sure most people share the same view. But the issue is that we have finite resources. And so if we could answer every possible scientific question to estimate every parameter with certainty, that would be great. But we can't do that. So from a funding perspective, we must allocate our resources to answer the right questions and to answer them within the right amount of certainty. Because we don't have unlimited resources, and some questions may not provide us much value to answer those questions. So moving on to vaccine policy recommendations in Australia. The way that we go about this is we have the TGA. the Australian equivalent to the EMA in Europe, to the FDA in the United States. A vaccine company will come along and apply for their product to be approved, and then it'll be approved by the TGA. We then have ATAGI, which are like a technical group. It's the equivalent to the JCVI in the UK and ACIP in the United States. They're sort of like, say, a technical group of specialists who make recommendations based on the evidence, and they will make recommendations to the Australian government and say, look, we think here is our advice on vaccine policy. These recommendations, if approved by the National Health and Medical Research Council, similar to your NRC, would then be implemented in the Australian Immunisation Handbook, and that's the equivalent to, I think in the UK, they call it the Green Book. Fortunately for myself, in terms of keeping my PhD feasible, they actually explicitly and transparently say this is exactly how we make our decisions. They use what they term the grade methodology. It's quite popular these days for developing recommendations and guidelines, and they get technical support from the National Centre for Immunisation Research and Surveillance. The way they do this is they set a policy question of interest. They use the PICO framework for that. So they explicitly define what they're interested in, independent of what research exists. They then do a systematic literature search. Mostly they'll find clinical trials, mostly as a product from vaccine companies. Sometimes there'll be publicly funded clinical trials in there. Sometimes it'll be supported with observational research if there's a lack of clinical trial evidence. Then what they do is they go outcome by outcome and they rate the quality of the evidence or the certainty of the evidence. So in that PICO question, they'll list all of the outcomes they're potentially interested in. So for vaccines, it might be adverse events. There might be immunological outcomes like antibody concentrations. There might be infection, hospitalizations, mortality, outcomes like that. And for each outcome, they'll go through and they will look at all of the evidence, all of the potential sources, and determine if it's low or high quality of evidence. Then they'll aggregate that in some informal way and determine some overall grade for this potential question. Once they do that, they'll then make a recommendation. Sometimes when there's insufficient evidence, they'll make a recommendation which effectively says, we don't recommend one over the other. So that they're making some sort of recommendation no matter what. So this method is used by the NHMRC in Australia. That's what they recommend for all of their guideline development. The WHO and the ACIP in the United States do this as well. For each outcome, they'll have a look at these 4 criteria. Risk of bias, you can think of as just, say, selection bias, or if there's a concern about confounding. Inconsistency will be if you've got multiple sources of evidence. They're all estimating the same effect, but they don't really align. You know, I think there's a high risk of infection versus a low risk of infection. Indirectness is usually when the research question doesn't map to the policy question very well. So, for example, this clinical trial looked at a 2-dose schedule for the vaccine, but I'm interested in making a recommendation for a 3-dose schedule. So I'm having to sort of indirectly infer that evidence quality. And imprecision is as you can imagine, what is the uncertainty around these parameter estimates? Based on these criteria, a grade is determined from very low up through to high. RCT data normally starts at high and then it can be downgraded for each of these categories. Observational data starts at low. There is a potential for an upgrade, although it rarely ever happens. Usually they're downgraded for various reasons. So as the first output of my PhD project, I summarised the assessments across these different vaccines and disease areas in Australia. This is what was publicly available at the time. I should note that it's only really been implemented for about the past 4 years. So there are a lot of policy recommendations that didn't use this method, but these are the ones that were publicly available. And perhaps not surprisingly, over half of the actual questions received a low or very low assessment. I should note that that is not the quality of the scientific evidence, it's the quality of the evidence to map through the actual decision making. Some of the common reasons were there was potential confounding. A lot of the questions, they only had observational data available. A lot of the time there was a mismatch between the policy question and the study-specific research question. In this case, it happens a lot because, say, if you're a pharmaceutical company and you're designing a trial, if your goal is to have approval in a particular country, usually the United States, because that's where the market is, you're going to be ignoring some of the populations within Australia, such as Indigenous Australians, where if the Australian government wants to make a particular recommendation for Indigenous Australians, they can only do that if there's data from Indigenous Australians, who often don't feature in these trials. And then, of course, there's uncertainty in the effect. And this quite often happens because we've designed the trial to assess this primary outcome, and that's how we've powered it and determined our sample size. And by definition of secondary importance are the secondary outcomes, but these aren't necessarily of secondary importance to the decision maker. They were just of secondary importance to the trial design. And so for certain outcomes, we'll have high certainty. For others, such as usually effectiveness outcomes like hospitalizations and mortality, we have quite low precision. There's just I've got two slides just summarizing the quality of evidence here. You can see that there were the disease areas, one per panel. Within each disease area, they had a different number of questions. For example, there was only one recommendation to be made for cholera, but there were 6 for pneumococcal. And then within each question, there was a different number of outcomes of interest. So, for example, for influenza, there were a lot of different outcomes that we're interested in. But for some of the others, such as rabies, there weren't as many. And the quality of the evidence varied quite broadly across the different disease areas, and then even across the questions. And so, for example, if you look at meningococcal, there were 4 questions. There were about 6 outcomes for each question, but almost all of them were rated very low or low, usually just because of scarcity of the evidence. And then if we look at the reasons that certain questions were downgraded, you see that they tend to cluster. So, for example, if we look at pneumococcal, that's the example I was saying before about the specific recommendation for Indigenous Australians is they were all downgraded for indirectness because there was no data for Indigenous Australians in these trials. So how could we possibly improve the quality of evidence for vaccine policymakers in Australia? Well, we could try and align the trial design with the policy question in particular. So, for example, you could have mapped the interventions and schedules. Of course, to a certain extent, we try and do this anyway. We do have investigator-led trials in Australia that try and do this. We could increase the inclusivity with respect to these particular populations. Something that I would like to try and do at some point in the clinical utopia that I see is to actually involve the policymakers beforehand in setting these questions and then trying to generate the evidence after the fact, instead of what we sort of do now, where for many different restrictions is a policy recommendation's made, and then we try and collect the evidence, and then we try and do this awkward sort of, well, hopefully the evidence backs up the policy recommendation, and then to a certain extent, sometimes we can't even do that. And then we want to generate evidence that actually informs the policymaker's decision as efficiently as we possibly can, and that's sort of where we sort of move into this innovative and adaptive design space. So a potential solution, I've called it the value-driven adaptive design. A couple of the other good names are already taken. So this isn't a method that is in any sense an optimal method, but by a series of metrics, which I'll sort of talk about later on. I'm just wanting to get people thinking about how we can potentially change the objectives of our clinical trials. So what if we knew exactly what a decision maker needed, and if we did know that, design the trial with the primary objective of answering that question? This is a big leap, but if we could potentially get decision makers to specify the function that represented their decision making, of course, humans are largely irrational, and it may be difficult to actually do this, so it would be nice, say, to sit down with a target and say, tell me exactly how you're about to make this decision before you see the evidence, and then we'll collect that evidence. Maybe easier said than done. And then we could design a trial to just collect enough information that sufficiently informs that decision. In this particular sense, I'm no longer concerned about type 1 error and power, because I'm not actually making a declaration about a vaccine superiority or treatment superiority. I'm more interested in reducing the uncertainty around particular parameters, which then feed into this function that the decision maker specified for their decision-making process. And perhaps if we do this, we could bridge the gap between research and translation and try and get that to map a little bit easier. So just some notation for how I'm going to go about doing this. We've got D, decision options. Theta is going to be our set of unknown parameters. Lambda is the willingness to pay parameter. I'm very much moving into the health economic space here. So the willingness to pay would be determined by a policymaker. So in the UK, you can imagine NICE specifying we value one quality adjusted life year at between £20,000 and £30,000. In Australia, you might have, well, I mean, it doesn't necessarily need to be quality adjusted life years either. It could be, this is how much we value saving one hospitalization or potentially even one life. It always makes people nervous when you're moving from like an effectiveness scale to a cost scale. But at the end of the day, that is how the policymakers act. So we need to acknowledge that. And then we might write down some net benefit function, which is really the willingness to pay multiplied by the benefit of a particular decision. And then you'd subtract the cost of that particular decision, because you can imagine different vaccines or different treatment options some are going to be more expensive than others. And when it comes to decision making, we're making that trade-off. So what we would like to do is, given this particular function, we then just choose the decision option that maximizes it. Typically, we'll reformulate this into an incremental net benefit, which is just the net benefit of one option minus another option. Usually you do that because it's easier to think about, but there's also computational advantages because you're reducing the dimensions by one. And so in this particular case, if the incremental net benefit is positive, you would choose decision A. If it's negative, you would choose decision B. So just as a quick example for this, in practice, you don't ever really know the exact values of these parameters. There's uncertainty through a distribution. So for example, let's just say we had two parameters, theta A and theta B. They're both normally distributed with their uncertainty controlled by this sigma parameter. If we visualized the distribution of the incremental net benefit, you can see how as the variance decreases, so as sigma decreases, naturally you're going to have a more precise distribution. The probability mass either side is the probability that one decision is better than the other decision. And so over time, you can see how the probability mass, is going to center around the true value, and we're going to be more likely to make the optimal decision here. But if we were forced to make a decision now, given our uncertainty, a rational way of doing this would just be to take the expectation over the parameter uncertainty theta, which is given by that red line there. In this particular case, our decision doesn't change depending on sigma, because it's always on the right-hand side of zero. But we can trust that decision more as we reduce the parameter uncertainty. Now, it costs resources to reduce uncertainty to actually collect data and run a clinical trial. So the obvious question is it worthwhile reducing the uncertainty, and at what point does it stop being worthwhile? So if we were to propose to collect data X, say run a clinical trial, we might consider the expected value of sample information. This is a classic value of information method. Basically, you have the expected value of our current decision. So given the current uncertainty in theta, we just choose the decision option that maximizes the expected value. We then have this term here, which is a bit more complicated, but basically we're saying, given all of the potential data sets we could observe, if we did so, and then conditional on that data, reduced our uncertainty in theta, so the theta given X is just the posterior distribution, then could we, what would the decision option be that maximized that? But we need to do that over all potential data sets, X, which is of course, in any reasonable case, going to be infeasible to calculate. Actually computing this over the past probably 20 years, there's been a lot of development in the approximation methods to actually do so. So it is usually feasible to do this within a realistic time frame these days. The way you would conceptualize that is basically the value you are going to get in expectation by conducting a study. And then if that value exceeds some cost of actually collecting the data, then that should justify collecting the data. If it doesn't, then we're actually better off just stopping now and making a decision based on the current information. So this is what it may look like for different sample sizes. You can imagine as the sample size increases, you're going to have more value given to you in expectation. But that's going to tailor off at some point. And you have an asymptote here, which is the dashed line, which is the expected value of perfect information, which is basically just, what if we knew theta with perfect certainty? How much value would that give us? And so you can imagine it goes like that. And it sort of makes sense that, especially if you start with something like a weakly informative prior, we don't know anything now. As soon as we start collecting data, we learn a lot very quickly, and then incrementally we keep learning, but we start learning at a slower rate. The blue line there is then our research cost for a clinical trial that usually costs a lot of money just to get it going. There's a lot of infrastructure you need to put into place. And then you can imagine you're going to have something like a per participant recruitment cost. So it's going to cost you more over time. So if we're just going with a fixed design, what we might do is choose n that, say, maximizes the difference between the value provided in expectation and the actual cost of collecting that data. But what we could do is something like an adaptive design where we're recomputing the expected value sample information at specified analyses. The reason we might do that is because, well, as with all sample size calculations and simulations and everything we do before a trial, it's based on a set of assumptions. We usually make assumptions about the data that we expect to observe. Hopefully they're accurate, but once we start observing that data, we might want to change course. One of the reasons why we do adaptive designs in general. But in this case, it's the same thing. The expected value sample information depends on our unknown parameters, and it depends on the data we're going to observe. So picking that fixed value may not be appropriate. We may want to update this over time. And that's really exactly what this idea is. It's as simple as that. So just some more notation to move the value of information into an adaptive context is we might specify analyses J. We don't necessarily need an upper limit on the number of analyses. We will then have the number of participants we recruit at a particular analysis. That doesn't necessarily need to be the same. They don't need to be equally spaced scheduled analyses. Here I've just got a bar over NJ just to say at analysis J, this is everybody we've observed up until now. In the red, I've got the observed data. I think that bar is rendering all right. So up until this current analysis, what data have we observed? In blue, I've then got the potential data that could be observed between analysis J and analysis J plus 1. And then we can just put that into this expected value of sample information formula. I think it's fairly straightforward there. On the right, you've still got, given what we currently know, what's the value of information? Given what we could potentially observe between now and the next analysis, how much value is there? And then the difference between them would be the expected value of proceeding from where I currently am through to the next analysis. So for this design, the idea is to continue collecting data until it's no longer valuable to do so. So we can recalculate this expected value sample information at specified analyses and make stop-go decisions based on that. If we assume the trial has a fixed startup cost delta and per participant cost lambda, we can use that to compare to this expected value sample information at these time points. So to start the trial, we would need the first analysis to be valuable enough to commit to spending the infrastructure to start the trial and then to recruit the first N1 participants. From that point onwards, we just need to compare that to the per participant cost and see if it's worthwhile continuing. And then once we're done, we'll choose the best decision option and then we'll report out the results as we normally would for any other clinical trial. As a case study, to sort of try and ground this as an example, RSV is a virus. It's if you're not familiar with it, just think of it as similar to influenza. There's a high disease burden internationally and also within Australia. I think this is probably one of the best cases I've seen for genuine equipoise between two potential decisions, where in Australia it's genuinely unknown whether it's more cost effective to recommend maternal vaccination, which is vaccinating mothers late in the pregnancy. Hopefully some of the antibodies flow through to the infant, or treating the infant with this immunoprophylaxis, which is like a passive antibody treatment shortly after birth. At the moment, it seems like infant immunoprophylaxis is possibly more effective, but it is substantially more costly. So from a policymaker point of view, there's genuine equipoise on which one they should recommend as policy. In this particular case, I make a lot of simplifications here just to go through this proof of concept. But we could imagine that we're interested in the trade-off between the cost of the strategies and the effectiveness, specifically in terms of medically attended RSV events within 12 months. Of course, preventing RSV is going to have a whole series of other societal benefits. It's not just going to be with respect to this outcome. But you could imagine how it would generalize if we wanted to extend it. This is quite a highly simplified health economic model. Just going through the components one by one, this factor is simply there just to put it on the scale of 1 million Australian dollar units. There's usually a discounting factor that the health economists like to use. So they will say, well, this treatment, is reasonably going to be valuable for 15 years, but the value of clients is going to discount by 5% every year. And it's the idea of $1 today is worth more than $1 tomorrow. On average, there's 300,000 Australian births per year. And then at an individual level, there's some willingness to pay parameter to avoid a particular RSV-associated event. The parameters we're interested in is this difference between the probability of a medically attended RSV event. You can imagine how in a more complicated structure, if there are multiple outcomes that we're interested in, this function would just extend to include the multiple outcomes. And then you have the cost, which is the difference in the cost. So in this case, per individual, maternal vaccination is 260 Australian dollars cheaper than infant immunoprophylaxis. So there's really only the willingness to pay parameter will specify, but the PII and the PMV are the only unknowns in this context. So it's fairly straightforward. If we knew those values, we would know the incremental net benefit, and then we would make a recommendation. So in this particular case, you could just simplify it to this comparison between those probabilities. Usually, you would have a policymaker say, this is what my willingness to pay is. For this proof of concept, I've just set it at 5,200 Australian dollars, just so we can think of it in terms of, if the difference in effectiveness is under 5%, then I'd recommend maternal vaccination because it is a lot cheaper. If infant immunoprophylaxis is more than 5% better on an absolute different scale, then I'm gonna proceed with that treatment. And so you can immediately see how it's different to a traditional trial where we would just be looking at this effectiveness outcome, and we wouldn't consider these cost effectiveness implications, which a policymaker would. So if we were going to proceed and conduct a clinical trial, the idea would be to do so to reduce a parameter uncertainty that would then propagate through to reduce the decision uncertainty. In this particular example, we may randomize up to 1,000 mother-infant dyads to each strategy, so 2,000 in total. We're interested in this 12-month medically attended RSV event outcome. It's just binary in this case, just to keep things simple. We might have analyses after every 250 dyads per strategy, so after 500, 1,000, 1,500, 2,000. There's no need to have an upper limit here, but let's say that we do at 2,000. Reasonable trial costs may be $1 million to start up the trial, and then 2,000 per participant. In this case, I specify these weakly informative prior distributions, which basically have the probability mass somewhere between 5% and 20%. And then a binomial likelihood. So a fairly simple model here. The reason I've done that is because, we've got this nice beta binomial conjugacy going on. That makes the EVSI calculations extremely simple. But in principle, there's no reason why this model can't be as complicated as you would like. It could be some enormous hierarchical model going on, but just to keep things feasible here. I'll look at two different scenarios. The first is if we would want to recommend infant immunoprophylaxis because it is more effective. It's, in this case, 8% more effective, which exceeds that 5% cutoff. The second one is if they're both equally effective, and we would want to recommend maternal vaccination because it's a lot cheaper. So for both scenarios, well, we haven't observed any data yet, so it's going to be the same for both. The expected value of sample information, just to go to the first analysis, is extremely large. It's $338 million. We use Mark Strong's algorithm, the non-parametric regression method, to estimate that. There's a whole series of methods. Anna Heath and others have done a lot of work comparing these methods, and the conclusion is basically they're very much context specific, and it's going to depend on your health economic model and your Bayesian model as well. Now, given this, it exceeds the cost of starting a trial, which is $2 million, and recruiting the first 500 participants. So we certainly want to start. And so usually in these cases, you have an enormous potential value gain to start with. And to some extent, that is determined with respect to your current certainty about these parameters. So if you have a really weakly informative or even non-informative prior distribution, then there is a lot of value to gain to observe any data whatsoever. If we started off somewhat actually knowing the values of these parameters, then that 338 million will be severely reduced. And so there's a little bit to play around with there, but you can imagine in a fixed trial design, you can almost guarantee that you're going to start the trial, and it's going to depend on how much uncertainty you have in those parameters to start with. Here I have just done one simulated run per scenario, and I've just simulated data and updated the posterior distributions. So it's fairly straightforward there. Scenarios 1 and 2 in the columns, and the rows are just the prior distributions, and we go down through these posterior distributions as they're updated. It's of the absolute risk difference, which is a quantity that policymakers are going to be interested in. The dashed lines are the true values used for simulation. And so you can see we collect more data, we're going to reduce that uncertainty. That's fairly straightforward. This figure is a bit different because it shows what the incremental net benefit is. We've got uncertainty there. And as you can see, as we proceed down and we collect more data, it's going to shift to one side of this dashed line, which is at 0. That's our decision threshold. And so you can see in the first scenario, we lean towards the left. And so we would prefer infant immunoprophylaxis. In the right column, we move towards the right. We would prefer maternal vaccination. Now, these figures alone aren't incredibly interpretable. You can see how as the probability mass shifts, we're going to prefer one decision over another. The decision on if we should continue is determined by this information, but also the cost of collecting data. If we're at, say, the first analysis, if it was extremely cheap to collect data, let's say it was 1 Australian cent per participant, then although you're very sure of your decision, it's still worthwhile collecting information because it's so cheap to do so. But you can imagine another world where it's so expensive to collect information, we're better off just stopping now, even if what would traditionally be deemed an inconclusive trial, it's so expensive to run, it's actually cheaper just to make a decision, even if it's the wrong decision, than it is to collect data and make a better decision. In these particular simulated runs, you can see we start off with a lot of potential value to gain by the first analysis. We do that. Then at the first analysis, we make the decision on if we should continue to the second analysis, which we do, because both of those values exceed $1 million with rounding error, scenario 2. Then by the second analysis, it's no longer cost effective to continue. So we stop at analysis 2. So you can imagine the 0.4 million is, at this point in time, the expected value of proceeding to analysis 3 is 0.4 million, but it cost us 1 million to actually get there. So it's going to be more costly to proceed. So we stop now. Of course, this is just one simulated run, and you can imagine a full simulation study around this. If you cast your minds back to one of those original curves that I showed before, you can see that the most valuable information to us is going to be the first pieces of information that we observe. And then we're going to reduce our uncertainty quite dramatically, and then it's going to taper off over time. So a current state of where this sort of field is up to, I had a meeting at University of Sheffield yesterday with Laura Plyce and Steve Chick and Martin Foster and Alan Brennan, who are listed here, amongst others, and that was really useful. There's a lot of literature developing these decision theoretic methods for optimal selection in a whole series of different contexts. Most recently in this space, there's this value-based sequential design that Steve Chick and the others have developed and they've published as recently as July, publishing a tutorial on how to do this. And it's been applied retrospectively to a series of UK-funded trials to see, what would have happened if we used this design. To my knowledge, I don't think there are any trials that have been developed prospectively with these methods. That would be really interesting. I've got a lot of interest in further discussions with this group and potentially some of this group here, just to see where ideas overlap. At the moment, I'm really sitting in a space where I feel like the motivation is solid, actually implementing it and seeing how these things pan out and determining, distinguishing optimal designs from suboptimal designs and then seeing, okay, pragmatically, how much of a difference does that actually make? What I'd like to see is in Australia actually trying to implement designs with these methods, but I've got 2 years left of my PhD, and as people keep reminding me, like, you can't solve everything in the PhD. You have a whole career ahead of you to sort these things out. In summary, I try and stand by the claim that clinical research should be designed to inform decision making, especially in the context when that's what we're saying we are doing to receive our funding. I think there's a fundamental difference in objective between the way we traditionally design clinical trials and how we and this particular idea, where we're moving away from a hypothesis test and we're just looking at, I'm gathering information to reduce parameter uncertainty, which will then propagate through to reduce decision uncertainty to help you as the decision maker make a better choice. But there's, of course, that was an incredibly simple example. There's a ton of complexities to consider here. There's going to be many cases where adaptive design is just not appropriate because, for example, and potentially even in the vaccine space, if you recruit extremely quickly and by the time you observe any outcomes, recruitment's finished, then there's not much to gain here. But of course, the health economic model can be extended, the statistical model can be extended, and there's all sorts of other decision theoretic models to work in here. And that's it. Thank you.